here. And I have known Dr. Zhu for a long time too, but I don't know so much about what he's been doing. There's only one thing that I think we crossroads on our pathway to here. So he graduated from, he's the chief scientist officer in GenScript, which is a longtime sponsor for CVB, leading new business development. He has PhD from Stanford University with molecular biology and immunology. And later he got MBA from UC Berkeley. You can see the path that he is towards that way, that want to make anything that he think it's interesting and useful, make it into true, implement into something that we can see, we can touch, we can use. And he worked in clone tech for a quality, very solid 10 years. I was fortunate to work with clone tech too, but when I entered there, then he already went to another path to be a founder to, next one. To be founder of the Genentestics. So, well, even though he left there, and my boss told me there are about 2,000 products in clone tech, and I will be the coordinator for about 1/3 of it. I was so scared. So I tried to learn everything, to read everything, to get familiar to those results, those products. And wherever I go, I always see his trace in there. This is developed by Dr. Zhu, that is developed about this, that. The most important system he pioneered and commercializes the yeast tube bridge system. Whoever is working with protein-protein interaction or protein-DNA interaction would never forget about that. It's very useful and very commonly used. And then he is the founder of that. After a few years, he returned back to China to Shanghai Zhangjiang High Tech Park. Now he's working as a chief strategy officer in GenScript. What caught my attention is not his education. He's working from this company to that company. His invention. What caught my attention is he is one of the unique scientists who always have something a little different than others. When most of the scientists, when we find something, we design a project, we get good results, we publish it, that's all. Then we have, we go up on that result, go find more stuff, get it published. It's a pattern like that, but he is not. He sees something, he wants to implement it, he wants to make it true, make it happen, make it into a useful product, make it commercialized. So when we say a scientist with business sense, doesn't mean a scientist with money in his sense, with the whole process. So we are very fortunate to have him here. Let's welcome Dr. Li Zhu.
Well, thank you. Thank you very much, Shuming. Do you see my image? Somehow I don't see myself.
Let's see, I will open the...
Oh yeah, start my video, okay.
Okay, yeah,
oh, okay.
You're there.
All right, okay. Again, thank you very much, the CBA, for inviting me, along with Dr. Linfa Wang. Yeah, I just want to tell you the truth. You know, today Linfa is the chief speaker. I am going to be, you know, the so-called pager, because he invented a very exciting system. Now I believe this is a very, very innovative tool for combating COVID-19 pandemic. So we picked this system from him and try to commercialize worldwide. So that's my mission, okay? But on the other hand, Linfa and I, we were actually, and Shuming as well, came from the same university. It's East China Normal University, Huadong Shifan Daxue, many years ago. Linfa and I actually were in the same class, right? And so we being classmates and friends and colleagues all the way 40 years, right? And then for such a long time, we connected each other because we know each other. But he mainly was in Australia and Singapore. I was in US and China. So, you know, until early this year, specifically, I would say in March, he told me he has something exciting and important. And then he was actually, I believe that he filed a patent the second day. He said, yeah, there is such a system I would like to commercialize and through GenScript. So this is the story to start. So far, it's been like 5 or 6 months and work together very, very closely. So again, I will just pick up where Linfa is dropped, so-called SVNT system, and to tell you what it is. Okay, let me share my screen.
Dr. Zhu, okay, sorry.
Yeah, I need to share my screen. Do you see anything from my screen?
Not from me.
yet. I think to click on share screen at the bottom of your screen. There's a green button.
Yeah, I already
then you need to click on what to share. Then in the new window, you choose. Okay.
Yeah.
Okay, here we go.
Yes, good.
Okay. Well, let me start from the beginning. Okay. Where's the...
在中间那个幻灯片放映就是上面。
This is it's a different from the.
Yeah. Do you see the red one? It says 开始.
Yeah, the play button. Yeah, this one.
Oh, okay. All right. So.
Okay.
Do you see this? Oh, okay.
Yeah.
All right. So I won't use too much time. Probably 15 minutes should be enough. So the title is this is called the SARS-CoV-2 Neutralizing Antibody Detection Kit. We call it the CPAS, this COVID-19 pass. Okay. Actually, from time to time, you will see different names. I think people attending today's webinar are all more or less concerned about this COVID-19 pandemic. This is a worldwide, affecting almost every single countries in the world, especially in US. I think the US is leading countries, number one in terms of number infected and the number people died. And then also, even after 9 months, starting from maybe Asia, but now in all over the world, US and Europe and Latin America and India, this is a wave after wave, still rising numbers. Like in the UK, now it's almost close to the second peak. So for the testing method for COVID-19, there are ways of doing RT-PCR, PCR, which is a nucleic acid test. And so far still it's dominant, whether you got a disease, right? That's the test. And then also there is some other called the serology test, IgG, IgM, ELISA kit. Those are the so-called, you take some blood and measure the binding proteins, antibodies. And that detect the binding antibodies. And then here is the new one. And the new one is, we call it the CPAS SARS-CoV-2 Neutralization Antibody Detection Kit. Okay. And this is actually the test for the immunity. I will tell you more why we believe this is the only one commercially available, and then also in terms of scale up, feasible detection method. So here's the commercial product. We call the CPAS, SARS-CoV-2 Neutralizing Antibody Detection Kit. And this kit is very simple, right? There's a box, there are some bottles there. There is a plate. Actually, you can do the 96 plate assay simultaneously. And the formula is very much like an ELISA. So the ACE2 protein attached to the plate, and HRP labeled RBD, which is S1, the SARS COVID-19, the membrane proteins binding domain is used for detection. You're trying to detect the interaction between those two proteins. And then this can be used with serum and plasma samples. The kit contains with positive and negative controls. Out of 96 wells, actually, the maximum you can do 92 tests with 2 positive and 2 negative controls. Then let this figure tell you a little bit more about how this detection system works. The COVID-19 virus that come to infect people or say respiratory tract. The virus, if there is no neutralizing antibody, they just bind to the receptor and then they infect, right? But if you have the neutralizing antibody, which can block this interaction and block the infection, and the same way is mimicked by this system in vitro in a very simple way. So here again, the conventional method already mentioned by Dr. Wang use live virus and use human cells. So you are doing this live virus infected human cells in a laboratory safe level 3. BSL-3 laboratories to do this mimicking infection in vitro. And then if the virus infected the cells, you form, say, plaques, right? The cell death. However, if you have neutralizing antibodies, then this plaque number will reduce, right? So that's called this VNT, virus neutralizing test. In some other ways, PRNT, plaque reducing neutralizing assay. So this assay is the best one because biologically it's relevant, but it has to be done in the BLS3, and then also takes a long time. The assay itself, just visualize how many plaques you read, detect. So it's not that easy. It's actually its accuracy sometimes very, very questionable. The method invented by Dr. Linfa Wang and now commercialized by GenScript, we call the CPAS, neutralizing antibody test. And this is actually ELISA format. So it's called the surrogate virus neutralizing test, or SVNT. So in this system, there is no virus, okay? There is no live virus, there's no live cells. All you have is the HIP conjugated RBD. That's a part of the virus membrane proteins as well, right? And this one can bind to ACE2, which is blocked onto the plate. If there is no neutralizing antibody, you will see the binding RBD to ACE2, you read 100%, right? Let's say. However, if you have the neutralizing antibody, the binding will not be 100%. It could be 99%, could be 50%, could be even 0. It depends on how strong or how fully block this neutralizing antibody can do. So this is a simple, fast, and can be automated. It works in any laboratory settings, can be done at high throughput and more consistent. Okay, so this is I would say this is the perfect mimicking the live virus assay, but much more faster, easier, and more accurate, economically feasible. So again, I think here I compare PRNT, which is a plaque reducing neutralizing test, which is considered gold standard. It uses a live virus. It's difficult using BSL-3 and takes 3 to 5 days. And then SVNT is the other way. On the right hand, it's a biochemical assay, and it does not need live cells or virus. It is easier and faster, less expensive. In the middle, there is called a PVNT. That's called a pseudovirus. And people use this in between the PRNT and an SVNT. But this one is actually not easy as well. Okay, that I use a pseudovirus like a lentivirus, which is spiked with say S1 or RBD. So again, I use live cells, just not using the COVID-19, and still it takes us 2 to 4 days, and then it's still you read this as like a plaque or some other ways. So it's not easy. People doing this when they do the few dozens already very tight. If 100, it's very, very tedious. So the only feasible is SVNT, which is invented by Dr. Wang again, and commercialized by Squid. So what's the advantage compact to this SVNT or CPAS with the some other people are doing so-called IgG/IgM ELISA kit, which is easy, right? There are a number of those commercial products available now in China, in US, in other parts of the world. Big diagnostic companies like Roche, Abbott, they all have those things. However, there's a big difference. You know, the binding antibodies, they bind to, say, target, which is S1 or IBD, but they may or may not be able to neutralizing it from binding to ACE2, okay? So that's the difference. The only way, actually, either you're using so-called gold standard VNT or our system that really to truly detect the neutralizing antibody. I'll give you some examples. So before that, I just want to tell you the serology test actually now has a lot of applications. You know, for herd immunity, immune passport, therapeutic validations, vaccine efficacious studies, all those things. Ideally, you need some very convenient, very efficient way to detect neutralizing antibodies. And that is our product can apply to. So there are several published papers. One is that this is actually USS submarine Theodore Roosevelt. And people probably learn this through news. In the submarines, 220, they got ELISA positive, but only about half of it had a positive neutralizing antibody test results. So there's a big difference, okay? You got antibody, it doesn't mean you have the neutralizing antibodies, okay? And you got a neutralizing antibody means actually you have the immune protection. I'll tell you why. And here, more studies. This is 2 studies done by European groups. I think this is one is testing, say, you know, if you're just using the total IgG, you see after 2 months or 3 months, the binding activity actually dropped significantly. Okay, and which is people, you know, feeling sad, say, hey, if we develop a vaccine, then the antibody does not protect it for more than 2 or 3 months, then what we can do. But if you're using the neutralizing testing system like a VNT or virus, or the best tool now is the CPAS or SVNT, the results are different. Actually, the titer does not drop that fast, that significantly, using the method to detect neutralizing antibodies. So you don't have to be that pessimistic. You know, I myself is more like optimistic if you have the right tool to study this. Then another story, this is done in Europe by this called Inatus in the Netherlands. Again, they take samples from a small town, which is infected quite heavily in early like March, April this year, and took those blood samples and test their first months and the second month and third month. If you test the total antibodies in the left and the middle, in those two figures, you see NCPS1, those are total antibodies, actually unfortunately dropped. However, SVNT, which is the kit they used to detect neutralizing antibodies, and they stay quite stable, at least 3-4 months. So I think this is just tell you the SVNT, similar to VNT and some others, which can tell you who have been infected. Anybody infected, you got antibodies. Those antibodies could be binding antibodies, could be neutralizing antibodies. But the only, do they gain a neutralizing antibody? You cannot tell by total antibodies or IgN, GM. But you can tell by SVNT. Does those neutralizing antibodies stay long? That's a key question. And I believe so far the evidence we have seen is if you're testing specifically neutralizing antibody, actually they are relatively stable. And I think there is another story, which is I can tell you, I think this is probably the best so far, the single case studies reported. And some of you may already heard of it. This reported New York Times. The story is this, in Seattle, which is also along the US West Coast, there is a boat in May. They went out for fishing, okay? And on this boat, there was about 120 people there. And then after 18 days, they are on the ocean. They got some fish, but they also got infected by COVID-19 because there is one single person before boarding actually carried the virus, okay, which is most people in the boat, they don't know. And so out of this 120 or so people on the boat, more than 100 people got infected. When they on board, they are PCR positive. Okay, so unfortunately, this is this is just a massive infection in such a isolated, enclosed environment. Most people you cannot just get immune. However, the story is this: before they went out, they did some serology test using two methods. Okay, the one method using Abbott total people, there's 6 are positive. The 6 people are all positive. Hopefully they have the antibodies already. They should be resistant to the infection. But in fact, 3 didn't. Okay, so only 3 didn't get infection, and the other 3 got infection. However, those 3 didn't get infection. Actually, is positive for SVNT method detection. That is using our kit. So this is perfectly what I'm, consider, and I hope you agree. If say using other tools, you're just telling you, hey, you have antibody, but really didn't tell you whether or not you have neutralized antibodies. But our kit can be done, and in such a way you can tell, oh, okay, so you have neutralized antibody. And exactly those 3 people on the boat, 18 days, out of that, they're still okay. They don't get infection. I think this is the most convincing data study, but unfortunately, I don't think that this story can be reproduced again in any near future. You cannot put people in such a risk. But I think in the next wave, when vaccine is going to be available, and by that time, I think this is going to be the perfect time to test whether neutralizing antibody are truly protecting antibodies, which we believe so. Okay, almost done. So this neutralizing antibody testing system we called SVNT or CPAS, developed by Dr. Li Zhu, now already approved in Singapore by HSA, in Philippines, and then also we got CE mark in Europe, and now submitted to the US UA for approval for emergency use. And so hopefully in the next 1 or 2 months, and we got approval, and then we can promote this clinical more widely application. So last slide, just a quick summary. The binding antibody, again, I'm just trying to tell you, is not truly protecting antibody. The neutralizing antibody are protecting antibodies. And so research working on the vaccine, drug development, and or convalescent plasma treatment, the CPAS system is the easiest, fastest, most scalable, and least expensive way to perform neutralizing antibody test. This kit is excellent correlated with VNT or PVNT, which is a live virus or pseudovirus, but it's much easier to do, much more efficient.